From Microbe TV, this is TWevo, This Week in Evolution, episode number 81, recorded on August 24, 2022. I'm Vincent Racaniello, and joining me today from Salt Lake City, Nels LD.
Hey there, Vincent. Good to be with you from LD Lab Studios again. Live streaming again. Looks like we're up and running. I actually did a little experiment, so or maybe I delivered on a promise, which was I was gonna, give a little notice on Twitter that when the time we're doing this, and so.
Good idea.
Yeah. So hopefully one or two people caught that.
By the way, folks, this is the podcast on the biology behind what makes us tick. And the last time I saw you, Nels, was at ASV in Madison, correct?
That's right. This is the American Society for Virology annual meeting, where sort of speaking of evolution, the origins of TWevo was because of an ASV meeting in the years gone by. But
that's right.
Yeah, this was really fun. So this year's edition in Madison, kind of reemerging from SARS-2 hibernation, coming first. I think we did okay. It felt like people were pretty vigilant, respectful in terms of public health measures. There was no ASV super spreader event that I heard of, which was good. As virologists, you probably want to avoid something like that. But yeah, that was really a fun meeting. Great to be together for a little while. And that actually spurred our last podcast. So folks haven't caught that yet. We had a really fun interview with Louise Moncla, who's about to open her lab at Penn, and ran into her talk at ASV. I guess, Vincent, you and I were both hanging out at that session, kind of separate sides of the room, and both kind of converged on the idea that it would be fun to have her on TWevo, and that turned out really well. So how are things going out there? Is the summer starting to wind down? Are you starting to feel the
it's August presence? Excuse me, it's August 24th, man. Summer's over. Yeah, another week and a half. I spent some of it traveling, most of it here at the incubator. Let's see where people are from. By the way, we have Les moderating today. Thank you, Les. This is Wednesday, so I do 2 live streams on Wednesday. I do TWevo, and then tonight is the more intense one where we have we have 500 people and rapid fire questions, which we can never get to. And this one I'm allowed to ask where people are from. So Angela is from the west of Ireland. Cool. This is very cool. I know that Rima and Rima's in Nebraska, and Les are moderators in California. Who else do we have here? Yes, Claire is from the UK.
Welcome back.
Rima's from Iowa. Sorry about that. Don't want to make it wrong. MK is from Eastern Massachusetts. Simon is from San Jose. Maureen is from Ohio. Rune is from Norway. Cool. That's very cool. Ben is from Switzerland. We have another one from rural Ireland. This is just great. And as Rima said, this is the United Nations. It sure is, folks.
United Nations of Evolution. I love it. This is great. Welcome, everyone.
Here's, what flag is that, Nels? Can you tell? It's too small.
India.
Yeah, India. Hello, welcome. Yeah. Here we have Michael is from Rotterdam. B. Clark is from Vancouver. Very cool. Amanda, closer to home, Pennsylvania. And here's Lost Lab. Hey, Lost Lab, how's it going? Still in Seattle.
Good to see you again.
William is from Omaha, and C. Bryce is from Washington.
Welcome all.
Great distribution of global interests, and all interested in evolution, Nels.
That's right. We share that interest as well, and I'm happy to report our paper actually today is going to bring us across the pond. So this is from a group in the UK. Should we jump right in? Vincent, or do we have other business?
A couple more. Let's see. We got Pete from London. We have Jay from DuPont, Washington, another Seattleite. I'm heading to Seattle in November. I will be there for 2 nights. I have to go to ASTMH and do a couple of TWIPs with Daniel and Dixon. But if anyone wants to hang out, get in touch.
Yeah, good idea.
I'll be
I was
right
just
near
there.
You're just in Seattle, yeah?
Yeah, a couple weeks ago. So we closed down the lab for a summer break, actually, and I headed out to the Olympic Peninsula
Mm-hmm.
for a few days. Met with some family and friends. Got onto Bainbridge Island for a couple nights, and then took the Washington State Ferry. This is my favorite form of public transportation, Vincent. Is getting on
Mm-hmm.
a massive ferry, driving your car on there, or walk-on passenger, or
Yeah,
bicycle,
yeah, yeah.
going between the islands. We went from Bainbridge to Seattle on the way back to visit some more family, and then headed out. But yeah, that was what a great part of the country for sure. Big fan of the Pacific Northwest. So
We got Dax was from North Carolina, Frank is from Santa Barbara, and Lost Lab wants to buy me a beer. You can do that. I think the Wednesday, November 2nd, that's my free night. We can hook up, tell me where to go. I'm down by the convention center, so.
Fabulous.
Someone B is going to drive down from Vancouver. Oh, you guys are cool. Very good. That's a whirlwind. October's a whirlwind, so I have to go to. I'm going to ID Week in D.C. I decided to check out that meeting, and then I'm going to Hawaii for an immune recording,
Wow.
and then coming home after each of those. And then I'm going out to Seattle. So those 3 crazy trips.
Full calendar. Yeah.
Anyway,
That's
thanks.
happening. We're reemerging.
Yes, we are reemerging, and I mean, travel is tough, because you travel, you can't do other things, and I'm realizing I can't do everything. So maybe I'll get the travel out of my system and then settle in here at the incubator and just be very judicious with my travel, because there are a lot of things I want to do, and if you're on the road, you can't do them.
That's
At least
right.
not very
Well, and
effectively.
then also, I don't know how much headaches you've run into traveling recently. We've had a couple in the last few months with delayed flights, sitting on runways,
Yeah.
etc. Plane changes, crew delays, all of the above, and so that might start to curb travel enthusiasm as well. But
Well, we have
we will
somebody
we'll
from
see.
Turkey here.
Wow.
Here we go. Peter is from Turkey. Cool.
Welcome.
Welcome everyone, and let's launch into our paper now. Something a little different. No COVID today, folks.
No COVID today. We're moving. Yeah, we're not closing the book on that, as we all know. But we are starting to explore some more evolutionary topics here, and maybe just to say up front, I think we'll get to this when we hit the mailbag. But I appreciate I'm starting to get some more recommendations on books and papers, bringing in kind of all facets of evolution, and that's great. That's the goal of TWEVO, and of course we've had our attention has sometimes gone to the evolution of SARS-2 for obvious reasons, and some of the microbial systems, but we do want to sample broadly, and so we're hitting a topic. We don't have backbones in this case, no vertebrates today, but we're heading that direction up the food chain somehow, out the evolutionary tree. So the title of today's paper is Evolution of a Novel Cell Type in Dictystelia Required Gene Duplication of a CUD-A-like Transcription Factor. There it is. And so really, I'm excited about this paper, I have to say, Vincent. So this arrived or showed up recently in Current Biology, one of my favorite journals, and is open access. So feel free to follow along on YouTube, and you can even open another browser window, point yourself to this paper, do a quick search, go to Current Biology. I think this might have published in June, and this is available just as we're stepping through the figures. You can join us right along for the ride. So before we get into the science itself and sort of decode that somewhat complicated title, let's first lift up the authors. So we've got Koryu Kim, Zihaihu Chen, Jillian Forbes, and Pauline Schopp. This is from the University of Dundee, School of Life Sciences in Dundee, UK. And then also with some affiliations at the Institute of Evolutionary Biology in Barcelona, Spain. So we're moving beyond just the U.S. science topic here as well. And that's, I think that's really fun as we're kind of going around the globe here with the folks tuning in on the live stream to also, we need to recognize that science itself is international. And to try to bring in voices from all over the world. Okay, so the topic here, Vincent, is the evolution of novelty. Broadly speaking, there's a few themes, I would say. The evolution of multicellularity. How do you go from being a single cell critter into sort of gaining specialized cell types? Obviously, some multicellular organisms do this all the time. Others, like Dictyostelium, so this is in the title, Dictystelia, this is a genus of slime molds that have specialized these complex cell types, or these beautiful videos. I'm kind of, spoiler alert, I guess I'm giving away your science pick of the week, are some beautiful videos taken by John Bonner. And we'll return to John in this. But so this incredible biology where these so-called slugs of slime molds are under conditions where they become starved, get attracted to each other, and then form these fruiting bodies. It involves specialized cell types. So we'll unpack that. But the evolution of multicellularity is on the menu today.
By the way, Nels, I have to remind you of Twevo 11.
Yes,
with Nicole King, who's also interested in the evolution of multicellularity, and she uses choanoflagellates.
That's so right. Cool. Previously on Twevo, exactly. This was a really fun conversation you and I had on the early, I guess, in our first season, in a sense, of Twevo. Was on this exact topic. And so, yeah, this is another case where, these seemingly simple critters, depending on changes in their environment, will undergo these really interesting sort of life history changes or lifestyle changes, which can involve multicellularity. And so that's, So if you haven't gone back or if you're interested, check out that interview with Nicole King or the lab, Nicole's lab, and some of the more recent work. Really, really exciting.
So Pete tells us that Dundee's on the River Tay, a few miles north of Edinburgh in Scotland. Fantastic. Daniel Griffin was just in Edinburgh, and he just doesn't pronounce it right.
Easier said than done. I'm not going to make a case for correct pronunciations
But I didn't correct him. I don't want to correct him on his podcast.
No, fair enough. Actually, one of the former postdocs in my group, Matt Barber, is doing a sabbatical in Scotland, in Edinburgh, and so
Edinburgh.
Thank you.
Edinburgh. Edinburgh.
No? Edinburgh. Okay.
Yeah.
All these fun connections here as well, however you might pronounce it.
I know I can correct you now. You're not an MD, right?
No, I think that's right. Sort of a different culture here in some sense, right? Yeah, exactly. Okay, so one of the big, sort of specialization in cells, and this is sort of what's going on here, is the division or the sort of the separating of somatic cells that, like our skin or things like that, from germ cells, the cells that go on to be involved in reproduction in the egg and sperm eating, the formation of gametes, or in this case, spores from the slime molds. And so, that's what's going on here with Dictyostelium. We'll get into some of the mechanics of this, but sort of to just step back and kind of consider the big picture here. I think the big question is really, how do these new cell types evolve? How do you go from being a single cell critter, or even maybe a multicellular critter with identical cells, and then start to see these specializations? How do you change sort of the developmental programs in ways that start to add to this biological complexity, these new functions? And so this is sort of, I would say, kind of one of the fundamental questions, or the work of the entire field, sometimes the shorthand of EVO-DEVO, or evolutionary developmental biology. And so, if we go back to the title of today's paper, this is so the evolution of the novel cell type is involved a gene duplication event of a transcription factor. And so that's what we'll be talking about today. And transcription factors are, these proteins that will get into the nucleus, interact with promoters in ways that modulate gene expression. So a transcription factor might be involved in the regulation of hundreds of genes as that protein is interacting with the nucleic acids in a way with other sort of complexes of proteins that come together, and then bring in the RNA polymerase to transcribe genes that will then be translated into new proteins. And so you can, through transcription factors, you can start to play around with the biology of the genes that you express in different times and places in an animal or another critter. In this case, a slime mold.
Nels, it's interesting if you just duplicate. So this duplication just happens, right? Because genes recombination happens, duplications of DNA happens. In fact, all these errors happen because they can be beneficial, right? So you duplicate a gene in the case of the transcription factor, and now you have everything it's regulating is 2x, and so you've got two copies, so you can play with the second one. Evolution can play with the second one, right?
Yeah, that's the idea. And so the duplication itself it could be considered a mutation. It's a random event. I think in this case, it's a copy and paste that actually so sometimes you have these tandem duplications, like you're pointing out, Vincent, where it's sort of like, in the not in transcription, but in DNA replication, you stutter or you recombine, you make two copies from one, and it's called a tandem duplication because they sit next to each other in the genome. In other cases, in this sort of copy-paste mechanism, there's another copy that shows up in a whole different chromosomal region. And so we think of those events in general as being random. And then as you're pointing out, the selection might act on that. And if there's some way to sort of bring that through, or it becomes beneficial through some amount of time, then you might sort of start to select on these new functions. And that's exactly the idea here.
By the way, Nels, I just had a conversation with Cedric Fichot up at Cornell last week, which I'm going to publish on TWIV in a couple of weeks. Talked all about mobile DNA. It was just great. You'll love it.
I love it. Yep. Yeah. So Cedric used to sit just across the hallway from me here at Utah. Moved his lab to Cornell a few years ago, but we've had some collaborations when he was here that were really fun. A lot, thinking about the evolutionary impact of these mobile genetic elements, and these projects are going on. We're seeing some incredibly, I think, for me, exciting stuff happening in that space. I'll be, my radar is on I'll tune in to see that interview. Can't wait. Okay. So in this case, we have a duplication of a transcription factor. And we have a lot of questions here. So what is this really kind of like, what happens? What is the impact of this, and how does this then connect to these cell type differences? But maybe let's also take a minute to just introduce the system, the critter that we're talking about here, the slime molds, the Dictystelia amoeba. And so I think the authors do a really nice job of kind of introducing the reader to the sort of taxonomy here. So there's 4 major groups. Probably the most famous of the Dictystelia is a species called Dictystelium discoideum. This has been developed as a model genetic system over decades of work from multiple labs, and you have all of these sort of genetic tools that are really going to come in handy here as we step through the science. And what's really fun, I'd say, about this paper is that now this combination of both evolutionary analysis and experiments. And so here we're looking at sort of a gene's eye view of this transcripture. So like if we start there where you're circling the amoebozoa, this is a tree just showing the relationship between these species. And so we've got in purple these 6 group 4 Dictystelia, including Discoideum, which will be sort of the star of this science, of this paper. And then just below that in gray is a species, or a different genus, a species called Viola violcium. I'm sure I'm getting that wrong. That'll come into the experimental situation here in a minute. And then the outgroup are some much more distantly related amoebozoa. These are the Canthamoeba, Entamoeba, these critters that you find in ponds and crawling around in dishes and things like that, in sort of the kind of mad scientist viewpoint or something. And we're gonna really go into that tree that you've got up here in a minute, but let me just sort of set up the biology for a second, if I could. So the lifestyle of the Dictystelia are really interesting. So they, as we were kind of hinting at, they're free living single cells. When they start to starve, they actually send chemical cues to each other, chemoattractants, that signal them to come together into this multicellular fruiting body. And this is where the videos are just wild. So you can see these cells migrating, they come together. And then it's at this step that they differentiate, the purpose of these fruiting bodies is to get this spore, those germ cells, up kind of off the ground, and to somehow broadcast the spores to sort of a fitness benefit for reproduction is the idea. And so then the stalk cells, these are the somatic cells, they're actually kind of build this pedestal as the cells come together. And then the the fruiting body, the spores move up in this sort of sphere, like it's sort of the reverse of the big apple. What is it on the New Year's Eve when you drop the apple down? This is the reverse of that. The apple's going up to the top to be broadcast in Times Square of the slime mold equivalent of that. Okay, and so and actually it turns out that starving is a sort of can be a panic signal for many of these single cellular critters to kind of signal it's time to mate. As you're mentioning with Nicole King, actually the choanoflagellates sort of do it differently. When there's bacteria around, they come together to eat and sort of hang out and multicell. So it's sort of cool to think about how these environmental differences influence multicellularity and how it can have these sort of different outcomes. Okay, so now we're just about ready to move to the evolution, but just to define a couple of the important cell types here. So there are these so-called cup cells that the group 4 species have from Discoideum, the other 5 that we were just glimpsing at. And actually the other species just farther out the tree don't have these cup cells. They still make stalks. They have a very similar lifestyle. They starve, they make the fruiting bodies, whole thing goes up, but they don't have these sort of specialized cell types. And so that's kind of where the action starts with this paper. There was some recent studies where people isolated the cup cells. You can do kind of microsurgery on slime molds. They're big enough that you can have access to them, and sequenced all of the genes, the RNA, and then compared what do the cup cells express versus others. And so that started to narrow down the set of genes that might be unique to the cup cells. And so that's where the authors sort of pick up this story. And so if you go back to that figure that you had up, Vincent. So they're showing a tree here, that big round one to the left, sort of the main action on the screen now.
Right.
They went in with the idea that there might be some transcription factors that are duplicated. And so these are the genes encoding those proteins that regulate the expression of a number of different genes. There might be ones that are specifically duplicated in group 4, but not in the other groups 1 through 3 that don't have those specialized cup cells. And they got a candidate here. And so that's what's being illustrated. It's a little bit complicated to sort of weed through that phylogenetic bush or tree. It's now kind of in this round format. But on the kind of left middle, you can see this CDL1A, CDL1B. So we're into some alphabet soup here. So this is a CUD-A like transcription factor. That's the one that they sort of zoned in on. And what you'll notice is there's those 6 purple species. There's actually 12 genes that are close by to each other here. Whereas if we look at the other, and by the color coding, if you look at the other species, there's 2 blue ones, there's 1 red one, 1 orange one, 1 green one, etc. So that duplication is specific to group 4. So that's something happened since that split in the ancestors onto group 4 where that gene duplicated. And then all of the other branches here, they're interesting, but they're a little, I think, less kind of consequential for this story. It just gives you some context of the relationship between the other members of this gene family. There's some.
Where's the CDL? Biology. I'm not seeing the CDL now. So where the heck is it?
So go up a little bit higher and to the left. There you go.
Oh, here we go. CDL right there. 1A and 1B. There you go. Okay.
Exactly. So you've got 12 of these genes. So you would expect to have 6. So if we go to some other genes, you see 6 purple.
Yeah.
And that's each one gene for each species. And here,
so there's one set.
Yep.
And then we have the second set, which is duplicated here, right?
Yep, you found it. Yep. And so that's unique for group 4. It's also unique for this family of transcription factors. So they had their hypothesis that, so that you find this, you find these genes expressed, these transcription factors themselves expressed in the cup cells that are specific to group 4. And in this one case, it's a duplicated gene. So this is like this comparison, this comparative analysis, evolutionary analysis led them to the hypothesis that something about that duplication could actually explain some of the specialization, the origins of the cup cells in the first place. And so
that was the theory that this kind of mechanism would be responsible for making new kinds of cells. So they decided to test it. And so now they have the first bit of evidence. We have a duplication, and then we have to see if it actually is what they think it does, right?
Exactly. And so, so the so that's where having a great model system with genetic tools comes in handy, is you can actually take that idea, that theory, that hypothesis, and say, okay, if this is true, we should be able to disrupt that gene and then see a phenotype, a difference. Okay, so the question then, so now if we think about the biology, what might we see? And so there's a couple of ideas just because of what's known about the biology of these cup cells and how they contribute to the formation of this fruiting body. And so the first sort of well-known fact is that cup cells help to, that's sort of that globe of spores, help it to go up the stalk and get positioned at the top of the stalk, which helps the spores broadcast, is beneficial for reproduction. And then, then the second question, so that's the first question actually. And so to do that, what they did is they knocked out the gene. So you can go in and do a genetic manipulation, engineer interruption into the CDL1A gene in this case, and then ask, what do those, what do these guys look like? And actually, I'm kind of jumping ahead here. So I'm just looking now at the second part of the figure, and maybe
I want to show a cup cell before you move on. Let me just show folks what a cup cell looks like, right? Because this is, so here's the stalk, right? And it's already differentiated, but here we have an upper cup and an upper cup in reddish, and it's yellow, and then a lower cup in there as well.
Yellow. That's right, exactly. And then the green cells, those are the spore cells that sit in between the cups. So it's kind of coordinating that, and then that's what's helping it to get up to the top of the stalk here. It's really worth it to look at some of the videos. I mean, it's just you might look at that at the end. It's like a knockout. But we have this idea that this gene is important. There's actually a little more evolutionary sort of evidence that maybe also kind of facilitates this idea as well. So I don't know, can we actually pull up that figure one again, Vincent, and scroll
the phylogenetic tree?
Yeah, exactly. And there's a second tree there. There it is. Panel B,
right?
Panel C. And so here, they're actually just kind of zooming in on the that gene duplication, so the CDL1A and 1B. And then there's that, you see that red line that they're indicating,
right
going
here.
from, there it is. So they're using that to signify. So they ran some tests. They could compare these genes between the different species and ask for, are there patterns of evolution here? Are there changes? So is the gene changing, the codon's changing in ways that could be indicative that actually natural selection is acting? It's not just it's a random duplication, but then selection acts. Is there diversification? Are the genes changing potentially in an adaptive way? And so I won't go into the weeds on this too much, but the answer, they're presenting some evidence where it appears like there's, the answer could be yes. And in fact, there is sort of overabundance of non-synonymous substitutions. These are changes in the DNA code that mean that the amino acid is different in the protein shape, or some qualities of it are different. And that those kinds of changes are overrepresented relative to what you'd expect just sort of by chance, or by cases where there's selection to not change the function of the gene.
Just to be clear, these different branches are all the different kinds of group 4 dicties, right? Those are the different species.
Correct. Yeah. So they take the transcription factor gene sequence from each of the species, and each one of those becomes a branch on the tree, on this gene tree. Exactly right. And then the duplication is below. So, and all of those other species are not shown here, but the group 3s, 2s, and 1s, the blues, the greens, the reds, they just have one CDL1. There hasn't been a duplication in those ones. And so that will bring a really cool and useful comparison point as we step through some of the experiments here. Okay, so I kind of jumped ahead and didn't include, but that's there's two motivations here. So there's the duplication event, and then some signal or evidence that there might be adaptive evolution happening, what we sometimes call positive selection. And this is, they're looking in the right place. So reproduction matters for fitness, right? Kind of one of the things that evolution does is, like, you get genes go through time. They go from generation to generation. And so this is reproduction, and the sort of biology around it are areas where it's very common to see natural selection acting. Okay, so now they knock out that gene CD1A in the model system, Dictystelia discoideum, and they just look at them. So so first of all, these slime molds survive. They do okay. Life goes on even in the absence of this transcription factor. The single cells seem fine, the so-called slugs. They come together, and then they start to differentiate into those fruiting bodies. They start to get organized. They're about to send the spores up the stalk surrounded by those cup cells, but no, actually it doesn't happen. And this is in figure 2. So they've got some photos here. I think my favorite one might be in that panel C. So here, this is really cool, right? So yeah, so they're in blue actually. So there's the stalk that's kind of running down the middle,
right?
And then in the wild type case, this is with all of the genes intact. They're using a marker. So those blue cells are those cup cells, and that matches what you just showed us in that cartoon, right? There's some on the top in blue and some below. The spores are sort of sandwiched in between. And that's what you want is, if you're a sort of, self-respecting Dictys slime mold, you want to see your spores up there surrounded by the cup cells. Now, if we look at that mutant, we still see the stalk. You can highlight the tip, but that didn't happen. It looks like those spore cells are below. There's no blue signal there. The cup cells are not there. They're not making it. And so the stalks itself, the authors point out, looks pretty normal. And you can kind of see that maybe in panel B, where they're zooming in on the stalk there. But the spores don't get to the top. And so they do another nice confirmation here, or sort of parallel experiment, where they actually surgically remove cup cells, and they see very much the same so-called phenotype. It looks like the same as if you knock out the gene.
You can see in these phase microscope pictures that the wild type, they have the bulge of spores there, and then it's not there in the knockout. Yeah,
exactly. Yeah, sort of another view from above, looking down on some of these fruiting bodies. Yeah. Okay, so then they can, with that, so that's sort of a good first experimental confirmation. They're kind of looking in the right place, and have identified a transcription factor that looks like it's important for this specialized cell type. And this, I just want to pause and it's kind of rare actually in the field to go from that evolutionary prediction to sort of validating with these experiments. And so the authors, I think, make a nice point of that too, that with the system, it's starting to put some of the mechanics, or the actual, the actual genes underlying these processes, and starting to teach us about how this really connects all the way down through the genetic level, all the way to the molecular level.
I guess in part it's because this is it's a model system that's been well characterized, and we know the morphological stages, and you can do a gene knockout relatively easily, and then you can see phenotypic effects. And it's not a mouse where you can have pleiotropic effects, right? And you don't know what's going on. Here you see the effect of knocking out that transcription factor, right?
I love it. Yeah. So, and thinking about Nicole King, so she took choanoflagellates sort of from the wild, and then has spent a decade or more sort of domesticating these, making it into a model system, building some of these tools. But there's some real advantages to sort of, tuning your evolutionary questions to existing model systems. And this shows one of those examples. Then they can do all these knock-on experiments. So they can do, in the absence of that gene, that mutant, let's ask what genes are turned up and which aren't compared to wild type. And they actually see that there are 130 plus genes that are underexpressed compared to the wild type. And so that starts to narrow down here. So when we talk about that transcription factor binding to promoters of genes, here's a set of them, 130 or so, that are influenced by the presence of that transcription factor regulating this process. So I think we're in we're pointing in the right direction here. And then there's some interesting developmental biology that they kind of go into. And so so for example, the cells that usually organize the cup and are sandwiching the spores, okay, well, do those cells still exist? Even so, in the absence of that transcription factor, are there cup cells? They're just not organizing, or where are they? And so they take a look at this and actually do see some evidence for the they're able to use some markers of these cells and show that during that developmental process, going from the slugs, as all of these single cells congeal and then developing into the fruiting body, there's a intermediate process called culmination. There are all these great developmental biology terms here. Those cells exist, but they don't get organized. They don't. So the same way that they would in a wild type case, that's not happening in the mutant. And then they do another fun experiment here, is to ask, okay, well, when this is working, what does that look like? So they take the CDL1 gene, they fuse it with a sequence to a yellow fluorescent protein. This is from a jellyfish, YFP. actually, when you code for that and make the protein, then you make the transcription factor protein, and it's fused to this fluorescent protein. And now you can see actually which cells are expressing that transcription factor, and how do they sort of move through the process of development. And so and that is kind of confirmatory that they're in the right place, and this is the biology of that transcription factor matters. So you've got the cells expressing the transcription factor are kind of spread all around in the slug. As it culminates, they start to get organized and come together. In the fruiting body, you see sort of that blue signal we were looking at on either side of the spores. You now see that in yellow. The jellyfish protein is showing at the protein level that transcription factor is also in that location. And so sort of, kind of building the case, I would say, through also the sort of the developmental biologist lens here. So, on the right path, but here's where I think where the evolution comes back in. And this is sort of like, I think the one of the founding principles of TWeVo, where it was in this era of where you can do experiments. How does these conversations between evolutionary questions and experiments kind of go back and forth? So not just like, here's a hypothesis, let's test it. Here's a hypothesis, let's test it. And then what does that mean for the evolutionary process? Or how can we learn more taking that evolutionary perspective? And so this is where the comparisons come back in, and I think really push the paper kind of to the next level for me. And so they start making some comparisons. So let's think about that duplication, or the impact, the biological impact of that duplication. So there's this other duplicate in group 4. These are the CDL1B. So that we've been talking about CDL1A, and then now we're talking about the other gene product, presumably another transcription factor that's working. Has it specialized? Are they totally redundant? How is this sort of working? And so they begin with that comparison, and they just do a knockout, but with CDL1B instead of CDL1A. And they see what I think they kind of, this is probably my own language or interpretation, but kind of wimpy stocks. So the spores are doing okay. They get up and get organized. They get up the stalk, but the stalk itself is a little bit flimsy. And this is, they show this in some micrographs, I think in figure 4 of the paper. And so this is kind of, points to that it's not a completely redundant function. So if the knockout of CD1A and CD1B was identical, you would say, okay, you had a duplication event, but now those two genes are still encoding proteins that are doing the exact same function. What we're seeing here is an example of neofunctionalization. You have different outcomes from taking away those genes, meaning that they're helping to conduct the developmental biology of sort of different aspects of this process of building the stalk, getting the spores to the top. And so in the kind of evolutionary parlance, this is often referred to as neofunctionalization.
I love that word, Nels.
Yeah, it kind of sounds futuristic somehow, even for like
it's very good
historical field somehow. And then you can, this is the magic of a model system. You can keep just tinkering away. So then they do a double knockout, and they see that now the stock has severe issues. And so this, I think this gets at a little bit of the complexity. It's not all of biology. It's like, we would love that simple story of saying, okay, well, this transcription factor is doing this, this one is doing this, but it's really, they're working in combination somehow. And that kind of comes through in the really severe phenotypes in the stalk morphology, and the spores aren't getting anywhere. The cup cells aren't there. Really a mess when you have those double knockouts. They just sort of sit there like slugs. They can't execute into the fruiting body program. Okay, but so now we've looked at within a species sort of that duplication, that proposed neofunctionalization, and they have some cool evidence for that. But here's where, the evolution can take you even farther. So you have, remember from that first tree, you have all these other species that didn't duplicate. And so what happens if we mess around with that transcription factor, CDL1, sort of the quote unquote ancestral in the sense that it hasn't duplicated? And so they do that experiment, which I think was really kind of, took the paper to the next level for me. And so this is with that P. violcium. This is a group 3 Dictystelia, and has a lot of the same cell biology or developmental biology. So the single cells starve, they come together into, they culminate and form a fruiting body, but it has a slightly different morphology. It's sort of, I think they show this in figure 5. Yeah, perfect. So here's now this P. vio, or P. viola, like viola, vio is better. That means stumbling over the pronunciation. So the wild type, you see, it almost looks like this kind of extended fan structure compared to the photos we've been seeing. And so then they knock out CDL1. And, I think my simplistic view was that, oh, well, that might be like the double knockout, right? That you would end up with like a super wimpy or non-existent stock and without any spores. But actually, here's a kind of a twist to the story. It actually, it's messed up and you can see that, but it's not totally lost, right? And so you get weird stocks, but it's not as severe, for example, as the double knockout, which would have been my sort of naive hypothesis or prediction going into that experiment. And I think this points to actually it makes an important point, which is, I said, I was kind of trying to be a little careful with my language when I said that it's ancestral for the fact that it hasn't duplicated, but CDL1 in this other species actually is just as modern as CDL1A and B in the group 4s. And so, so evolution can work in many different independent ways. And so maybe there's been a neo-functionalization here with this other species that didn't involve the duplication of CDL1. Maybe there's another transcription factor that got recruited and now can cover for a lot of that stock formation for even getting the spores sort of semi-position. And so I think there's probably another story, an independent story about the specialization of those cells for that other species. And so that's this is actually one of these, I think, an important take home for me, or, if you remember one thing from the TWEVO, it's that when we're trying to compare sort of ancestral states, but with modern species, there's been just as much time. Like there's the coelacanth, the living fossil. It's a living modern species. It might look to us like a fossil, but it's had just as much time to evolve and to sort of dispel the idea that there's sort of among the modern species that there's more primitive or less primitive species based on sort of complexity or cellular features. We're all just as modern. We've gotten to today through different evolutionary pathways, but with sort of really cool, possibilities of different biology happening. I think there's probably another story hiding
sure.
in that species as well.
They're very lucky that these deletions are not lethal, right?
Yes, absolutely. That's
right. The organisms can grow to a certain stage, and they obviously have problems. But when a mutation is lethal, you learn less because you can't see, and the deletions here are giving us good information on functionality. So it's really good.
Great, really good point. And that's like your point about a lot of the challenges with mouse genetics is, and with developmental biology in particular, if you mess up a key developmental process, those mice aren't going to live to be adults.
Yeah.
So then, the field has moved forward by developing the technology to do a lot of conditional knockouts. It might be that you have that gene you need to get through a developmental time course, and then treating with a chemical or using some other genetic tricks, you then delete it later in the life of the animal, and then can ask, okay, what does this matter for this part of development or part of the adult lifestyle? But that's a lot harder to do and technically a lot more difficult. So that's an important point, that these experiments work in part because they're doing this sort of specialized part of the development here. I would say it would have been fun, so as we always do, Vincent, a little podcast peer review, and it would have been fun, I think, to see some of the fitness assays. What are the consequences of having these weird stocks? Are they as good at, if you take away those genes,
sure.
Are they as good at broadcasting the spores? Maybe it's kind of obvious. If you've messed up that structure so bad, then it's probably not going to be so useful. But I'm curious about that, especially let's say in the CDL1B mutant, where it looks like the spores still get up there. The stock is wimpy, but does that really impact fitness? We don't know.
I'm sure they have such assays that they do. That would be interesting to look at. I agree. Yeah.
Yeah. Kind of connects it to the ultimate phenotype, which is, are you able to reproduce? Is selection acting to favor reproduction through the origins and then elaboration of the cup cells in those group 4?
So Nels, how many amino acid changes distinguish the two genes?
Yeah, good question. And I will confess I don't have that number directly.
So they say there's evidence for positive selection at 7 different sites.
Yeah.
Right. So, there are a lot of other changes, but they don't seem to be positively selected. So maybe those are the functional one. So 7 is not a lot, right?
Yeah, no, exactly. So I think we're already kind of daydreaming about some fun experiments, future
Yeah.
experiments here, right? Which would be to sort of ask, what are the consequential mutations? So if you transplant those 7 back into, in either direction, kind of make chimeras
Yeah,
with
sure.
CD1A or B, and then say, and then C, does it start to go more
That would be
towards?
interesting to trace the development or the selection of these changes over evolutionary time, right?
Completely agree. Yep.
And
Yep.
so, what do you think? What is the time scale we're talking about here? So it duplicates at one point, and then it's identical. You have an identical copy,
Yep.
and
That's right.
then it starts to drift, and then at some point you have. So what? How many years does that take? Do you have any sense?
Yeah. Well, in this case, a long time, I would say. So I didn't do my background into group 4, the predicted time, and I don't think they say it, at least in the main text.
No, I
Predicted
don't.
sort of last common ancestor of group 4. That would answer your question. And then, and just to totally agree on those 7 changes. But just to point out, there's more changes actually. So those changes are highlighted because they have that signal of positive selection, meaning there's sort of multiple substitutions across the comparison of the several species. There could be 50 more changes in there depending on the length. And so, and sort of those signals of positive selection, there can also be cases where, one change that's shared by all of them could actually be beneficial as well. And so a lot of times in cases like this, with nice experimental tools at hand, people will not just like grab those 7 and make that chimera, but we'll kind of do it in chunks. So they'll say, okay, let's take the first half of the gene with the change, with the other duplicate, put those into and then, sort of start to march down the gene and then try to narrow in. It gives you sort of a full view of all of the genetic changes that could be important. And so, yeah, that's another way to do it.
So the knockout of the gene is actually not a fair experiment because the other copy has been evolving with it, right? And maybe they're compensating. So you pull it out. Yeah. It's not a fair question. It's not a fair way of saying what was it like without the duplication because the other copy has changed, right?
Agree. Yeah. So that's a different question, and a good one. And so actually, there are ways to tackle this. So you can start to do ancestral reconstructions. So you can, so instead of taking the modern copies and kind of messing with those, you can try to predict using the phylogenetic analysis that's already shown here. You can actually go back to those nodes and say, you can use algorithms to predict this is what we think the amino acids sequence looked like, the gene sequence looked like in that ancestor. And then you can ask, okay, let's do the double knockout, which they have. Let's try to rescue with that ancestor, and then ask, what happens? Does it make, what does that look like? And so, but I think your point, I love this direction of thinking, and it also, I think, very quickly you start to recognize, okay, hold on, well, we can't, evolutionary biology is pretty powerful, but it's not a time machine. We can't go back because right. It's the real question is, what did every gene, what did the whole network look like in ancient times? And so we're dropping in that ancient copy in an otherwise modern set of genes. And so you have to kind of keep an eye on that and think about the interactions, or the kind of technical term epistasis, that is going to happen when you sort of mix and match these things.
Because presumably at one point there was, this organism had an ability to distribute its spores, right? And so whatever happened after the duplication improved it in some way. And you'd like to go back to that state and ask why a different state was selected for after the duplication, right? Because it had to be better in some way. It's fitness, right? So
that's the sort of assumption for sure.
Yeah, that's assumption. It maybe not. Yeah. Maybe it's not all selection. Who knows, right?
But I mean, in the case of reproduction, there's a pretty direct line, and so you kind of you've picked a pretty good. You're kind of you're looking in the right place, I would say. But you have to be careful. You're right, because it's very easy. I think, especially our human brains are really tuned to see sort of adaptive. I mean, in some cases, is like referred to as just so stories, that it feels right, or it kind of feeds into our intuition.
Well, we're used to thinking that the trajectory of evolution is to get is to make a better machine, right? But that's not always the case.
Exactly.
Evolution makes something that's better for the moment. Not
or even equal for the moment, or just was pulled out of the marble pulled out of the bag at the moment, and it can be all of those things. That's right. Yeah, a really important point.
Yeah, so because it's for the moment, and then what happens in 10,000 years is something different. So don't look at evolution as trying to build a really good jet plane that can go long distances on no fuel. That's not how it works. And I was guilty of that years ago as well, of thinking that way, but it's not, because you look at a complex mammalian eye and you think, oh, this was the goal of evolution to make. No, it wasn't actually.
That's right. Yeah. No, and things can kind of get messy pretty in a hurry if we allow our intuition to sort of overpower like kind of these evidence-based inquiries as well. So so it could even sort of move into sort of philosophical or religious space. I won't dive into that one here, but maybe to close just on the questions ahead for this. So I think a really cool example of a duplication that had some real consequences, maybe more complicated than it first appear. But yeah, and I think you've already been kind of hinting at these questions, so how do you go from the initial, like that day it duplicates and they're 100% identical to, now flash forward a million years or whatever. And with this complex scenario. And so using some of those ancestral reconstructions, again, come with caveats and not perfect, but a way forward to think about that.
Yeah, I think they would. And you mentioned some of the ways that you could address that. And I think they're probably going to be doing that. Those kinds of experiments.
Absolutely.
Right?
Yeah. I'm curious to see what happens with these questions ahead. Also, how does like a common question of developmental biologists? So how does this all get wired together actually? Right. So we kind of, we show up
very slowly.
Yeah, that's right. Well, we show up and we also show up in the middle of the story or the middle of the biology, which is, okay, here's this transcription factor that's helping to guide the developmental process to make these cup cells appear at the right place at the right time. But what's telling that transcription factor to get there or to organize that in the first place? And so you can always kind of go upstream and ask, okay, well, what does that look like? Both sort of from this one snapshot of this one species, but also from that evolutionary view. How does this get wired together? And then downstream, those 135 or so genes that are turned up or regulated, how does that wiring again get connected or reconnected in the presence of the two at first identical genes? And then as they diverge away, how do you sort of differentiate between that? I think starting to do some of that same RNA sequencing, but with the CD1B mutant, is it the same 135 genes, a subset of them? Is there different genes? And kind of thinking about it from that perspective. They go into some of those details for CD1A, but I think adding that comparison could potentially be pretty powerful. And then comparing again to that close relative that only has one copy. Is it the same set of 135 genes or what subset? I think there's some really fun possibilities.
So Nels, obviously viruses would be a good system to study how duplicated genes lead to neofunctionality, right?
Yeah. Well, now we're back on our favorite topic, of course. Yeah. I mean, so this is the magic of viruses in some senses, that they're sort of masters of this genomic gymnastics. And so these events that might happen really more rarely in some of the critters with massive genomes and lower mutation rates and things like that can happen sort of in the blink of an eye with some of the viruses. And then I've been kind of thinking recently about, also these viruses, not only as sort of separate genetic entities, but as they're interacting with hosts, and then getting at your point or your conversation recently with Cedric Bichat, the mobile elements, and what is the genetic conversation between hosts and viruses, in some cases mediated by these things. So basically the virus genome could be a test bed for duplications, other things, but could, in horizontal transfer, could some of these genes be regained by hosts or sort of transferred back and forth, and how does that influence the evolutionary conversation? Yeah, no, lots of really cool.
I'm thinking of your work where you showed the amplification of a specific gene in vaccinia virus, right? But then you remove the selection and they go away. But what if you kept it? Would you start to see drift among the copies, and could they eventually acquire another function? Boy, that would be a cool experiment to do. It would take a long time. Right.
I agree. We've kind of started down that road. We haven't seen that, but I like that. We should add that to the menu. That's a really nice idea. That's exactly right. So these things kind of in the blink of an eye can go from one copy of a gene to a dozen copies of the gene, and then, seeing how evolution might tinker with that. So we've done a little bit of that. We see some cases where the genes sort of homogenize by recombination. So like if one mutation shows up that's beneficial in one copy of the array of like a dozen genes, come back next week and might be half of those genes have that exact same mutation. And it's not that they're independently gaining that mutation. It's that through recombination and during replication of the genomes, it starts to spread or homogenize, almost what a kind of equivalent of gene conversion is, it's been coined in other systems. Yeah, no, all kinds of possibilities for evolution to kind of go into turbo in the context of a virus as it's replicating.
I want to see if we go back and pick up some
questions. Yeah.
Some questions or comments here. First, welcome, Steph. Thank you for moderating, joining us today, and helping Les to moderate.
Cool.
There was here, Angela said there's quite an interest in slime mold macrophotography because they produce such beautiful fruiting bodies. Yes. That's cool. Yes.
We'll return to that. Good point. I couldn't agree more.
Andrew says one could try seeing how the stalks work in various environments.
Yeah, another good point. So I've kind of been taking a very simple view that, so this is all about reproduction. Like if you get your spores up a stalk, they'll go farther or something like that. But then really good point, Andrew. So imagine, maybe there's an environment where having a thicker stalk makes more sense, or maybe at an intermediate level makes sense, or there's all of this, like when you bring in the environment, you got to help bringing in the ecology could really unlock some of the evolution in the biology here. So,
Rob says, I think the knockouts would cause highly localized results.
Yeah, and that's kind of what you see. So the the idea that these things survive, they try to make spores. The spores don't quite get where they belong, or the stalks are a little bit wimpy. But most of the biology is in place, and that shows. I think it's a really nice example about So we've been talking about one family of transcription factors. There's lots of transcription factors at play, and some of them master regulators of key processes that could be essential for life. Others are really kind of tinkering with the other, bringing in a different cell type, and it's so that's on the money.
So Nels, how did the first transcription factor evolve?
Ooh, I choked on my tea there with that question. That's a great one. I think we're as we're seeing, and today we're out on so that's like the core, excuse me, the tree of life here in some sense, right? Where as this biology was just getting online. For this paper, we're out on the very tips of the branches. And that itself is so complex. But I think it's the right question. It's the right way to think about this is how can we use the knowledge that we gain from kind of starting at these modern states? How far back in history, how far back into the tree can we start to unravel how some of this stuff happened? And so, wow, that's an ambitious and awesome question.
Well, Eugene Koonin thinks that the RNA binding domain is one of the most ancestral protein motifs, right?
Yes.
So you evolve the ability to make protein, which in itself is a huge thing, right? I say it in one sentence, but it's huge. And then it's very easy to imagine how you can pull out from a mix proteins that can bind nucleic acids, right? Just have the right combination of amino acids, and then amongst those came the transcription factors, because that's what they do. They bind nucleic acids, right? And eventually you could see it coming from that, regulating and making more complex organisms and regulating gene expression and so forth. So I think it has very sort of clearish roots, but proving it is very hard.
That's the part that where it becomes really challenging. Intuitively, that all makes sense to me, and sort of hits on a theme of evolution is, so something, as soon as something's invented, in this case, so if we're sort of assuming an RNA world, right? And then the ability, so you start with kind of the ribozyme era where the RNA, you don't have to translate it into a protein. The RNA itself can fold in a way that it's catalyzing a reaction. But then from there, you bring it, as you're saying, you bring in the ability to interact with nucleic acid, to do something different with it, to make something. Now you could start to, once you've kind of got that foothold, you sort of borrow that machine to then do all of this other biology that's related to it. And so, that all sounds right, but it's exactly that last point, which is okay, now prove that. And
Isn't it? That's the saying. Nothing. Evolution doesn't invent anything. It just repurposes, right?
That's yeah. So, and that's, is that a cop out? It's like easy to just like, they're the things we can draw those connections and those lines.
Yeah.
At some point you have to like go out on a limb and say, okay, here's like a completely novel thing, to get at this. Yeah. To get either the biology in the first place or to,
And with respect to this idea of making a better machine, giraffes didn't evolve long necks to reach the top of trees. That's right.
Yeah, the old American idea, which,
Yep.
I love being open to any ideas, but where's the evidence and can you explain, is it easier or is the evidence more consistent with this sort of random mutation selection acting versus kind of a as you go adaptive process, the giraffe reaching the top of the tree? Yep.
Kang says, are there any implications for trisomy?
Yeah, I like this this direction of thinking, right? So how many copies of stuff that you already have will that influence? So here, these guys are diploid, meaning they have 2 copies of every gene to begin with, and then you're making a copy on top of that. If you have 3 copies of everything, there are critters that are triploid and up the ladder for number of copies, and that certainly can influence the ability of gene duplications to happen or the frequency of them to happen. And so absolutely, yeah, that can certainly become part of the story here. Trisomy, in a lot of cases, we're talking about aneuploidy, where it's not the entire set of chromosomes is in 2, 3 copies, et cetera, but there are regions that are in extra copies and how that influences. And so, certainly I think the ones that first come to mind are a different fungi, yeasts that where we see where experimental evolutionists have gone in, put selective pressure, and you very quickly see aneuploidy events where now 2 copies becomes 3, and very quickly that can add an evolutionary advantage to like scavenge some nutrient or avoid some antifungal treatment or things like that. So yeah, that's certainly this idea of kind of wholesale big genome scale changes or chromosome changes or parts of chromosome changes absolutely at play in a lot of cases. Here it's almost like the scalpel has gone in and just taken this one gene and made a second copy of it, a much more sort of modest and refined duplication event.
So Trion's been asking a bunch of questions about SARS-CoV-2, but Tryon, come back to the live stream tonight. We'll answer those. Some of them are pretty good questions.
Yeah.
But this one is relevant.
Yeah,
there are a number of drugs that can enhance mutation. I mean, molnupiravir is a mutagen. It causes the polymerase to make mutations, and that ends up inactivating the genome. So that's how it works as an antiviral. So any drug that gets incorporated into the nucleic acid and then causes the polymerase to mistemplate, right? It sees a base, it sees a U, and instead of putting it A, it puts something else in, right?
Totally. And drugs can also lead to duplication events, actually, or these aneuploidy events, right? So antibiotic resistance in a lot of bacteria, you put an antibiotic on them and they come up with like a 3rd copy of a transporter that can pump that drug out because there's so much of it get in. So it applies in many biological scenarios here for mutations, not just point mutations, as Vincent's pointing out, but all flavors of them.
Yeah. Okay.
Great questions or comments. Thanks everyone for
good questions.
Yeah.
We have a nice little group that appreciates the idea that we can talk about a paper and you guys can ask questions, and I love it. I think this is a great forum for that. And this is a manageable size. We have 63-ish people. It's a manageable size. It's like a moderate-sized class where if you have a lecture hall full of 500 students, it's really hard to have a discussion, right?
A fire hose of comments, so it can become overwhelming. Yep. Also, next time I'll not only sort of tweet our timing, but maybe put if we have another paper ahead of time, far enough ahead of time, put that in as well. So in case
I put that in the YouTube.
Oh, you did? Oh, fantastic.
Notes, which people can see. Yeah,
that's great.
Oh, someone just joined us from Shanghai. Welcome.
Oh,
cool.
Welcome.
It's Very morning, cool. Huh? Good morning.
Yeah. All right. Well, maybe we should move to the mailbag here.
Yeah, we do have a couple of emails today, so let's check those out. Yep.
I'll read the first one here. So this is from Bree. Hi, Dr. Aldi. Thanks so much for doing TWIBO. As a biochemistry postdoc, I find it really fascinating. I've been meaning to write for a while because one thing that I think about a lot when listening is whether the DN-DS ratio, so this is the ratio of non-synonymous to silent substitutions, we brought this up with the paper a few minutes earlier, whether the DN-DS ratio is missing important evolutionary changes that affect gene expression in related topics. So how do evolutionary biologists investigate that? I got up the courage to write when I came across this article, and I was wondering if you could maybe discuss it in an upcoming episode. Thanks for considering it, and thanks for the informative podcast. This is from Bree, who is in San Francisco. Bree, so thank you so much. We actually, so this paper really came on the radar. It got a lot of noise here. So synonymous mutations are in representative yeast genes are mostly strongly non-neutral. So, and this is from George Zhang's lab in Michigan. This really kind of caused a ripple in the field, at least on science Twitter and probably some other circles as well, because so this ratio, the DN-DS ratio of synonymous to non-synonymous to synonymous, and again, the non-synonymous changes, these are mutations, point mutations in the codon of the DNA that mean that you code for a different amino acid versus quote unquote silent or synonymous changes. And so because the genetic code is degenerate, you can make some mutations but still encode the same amino acid. And so the assumption in a lot of these models where you sort of compare rates is that those silent changes are going to be not impacted by selection. The protein will look the same. And so whatever, if you're recognizing a pathogen or it's involved in, recognizing a gamete or something like that, the protein will look the same. And so there won't be sort of an adaptive process there. And so that's how you sort of gauge whether you propose a gene is under positive selection, as the authors in our paper today proposed for CDL1A. So this is, the reason this is a ripple in the field is it's saying, well, actually your assumption is all wet. It's a mess because even the silent changes have an impact on, even though the protein it codes for is the same, maybe the level of the protein is different because the other machinery that's coming in and transcribing, et cetera, are having an impact here. So, and this has been pointed out before many times, and there are definitely cases where selection can act on synonymous sites. I think the reason this one kind of got a lot of attention was it was basically saying not only is this like a little effect, it can be a pretty big effect on fitness. And so all the experiments were done here in yeast. People kind of got up in arms for a week or two. Because that kind of takes a broadside at a lot of my lab included. Sure. It uses this approach a lot. And so a couple of other groups, thinking Leonid Krugliak, who's at UCLA, Craig Kaplan, who's at Pittsburgh, and a few others kind of tackled that idea. And so these are yeast genetics labs, and kind of took sort of post-publication peer review, took a closer look at how these experiments were designed. So how they were measuring or proposing the fitness differences from the experiments they set up as they engineered in silent changes, non-synonymous changes, tried to do a prediction across a number of yeast genes. And actually pretty quickly they found some potential artifacts in the way that the experiment was designed. And so this was sort of, there was this big Twitter dust up about, or, scientific dust up about, hold on, throw DN-DS out the window. And then a few weeks later, and a second dust up was like, hold on, actually, let's look at how this study was done. And so unfortunately, I think that so I've looked fairly closely at that, and I'm convinced that, I'm not convinced that the sort of the results or the interpretation of the data actually matches sort of what's proposed in that paper. So
you're talking about this Nature paper, right?
Correct. Yeah. Okay. So I think, that there are probably some technical issues with the experimental design that made it seem a lot more that the synonymous changes were having a much bigger impact, functional impact than than is actually going on.
But you can imagine that certainly secondary structure would have an impact, right? But it may not be to the extent they say mostly strongly non-neutral. Maybe that's too strong, but there are certainly going to be some synonymous mutations that affect RNA or DNA structure, and I totally buy that. That's fine.
Me too. Yeah, and there are. That's an assumption that silent changes are sort of quote unquote silent.
Yeah, of course.
And that's violated all the time. Sometimes I try to think about it as like the analogy of rearranging the deck chairs on the Titanic. So like that's like you can rearrange the deck furniture. That has an outcome. There's a functional outcome. But then if the Titanic sinks because a virus came in and wiped out the whole population, then those changes sort of go a little bit into like whether the deck chairs were kind of here or there is a little less consequential than the whole ship going down. And so, yeah, I think a lot of especially in the host pathogen kind of thinking, a lot of these like massive selective pressures for survival, also in reproductive cases, the strength of selection can be really strong. I mean, the viruses themselves have been teaching. SARS-2 has been teaching us this over the last couple of years, how quickly mutations can kind of arise, sweep a population, and then disappear as we're stepping through.
Yeah.
Pandemic. In that case, evolution can happen pretty drastically, pretty dramatically.
Well, I think the DNDS, whatever the flaw might be, it still will identify positively or potentially positively selected amino acids, and then you can study them and you can do the experiments to prove or disprove whether they're actually selected for or not in terms of the phenotype. So if you're thinking if it's a receptor-ligand interaction, you could actually look at it directly, right? So absolutely. I would argue the DNS points you in that direction. What you just can't do is throw out all the synonymous ones and say that they're never selected for, right? That's it.
Agree. 100% agree. And that's where having a model system or the ability to follow, like to use that kind of analysis for hypothesis generation, but then to kind of guide experiments to ask yes or no. And of course, the environment's going to matter that you're testing it in, the types of things, the phenotypes you're looking for are going to matter. And so it can get complicated.
All right, here we go. Here's our first joke. A pair of mouse twins are synonymous.
I love it.
That's really good, Pete. Very good.
And thank you, Bree, for reaching out. So Bree also contacted me, I think, by direct message, and I was able to. We had a fun conversation, pointing to some of these threads as then the specific details of that paper were analyzed. It'll be interesting to see. So I like George Zhang, the senior author here, well respected in the field, great evolutionary geneticist. I'm curious to see how, I haven't heard actually how they're responding to this, if they're modifying their conclusions here, or where they're sitting with the conversation. That would be interesting as well. Hopefully it's not turning into an adversarial scenario.
No, it should be a good discussion. That's how science thrives. Actually, Ben here has a good question. Wouldn't the outcome of knocking out the duplicate also depend on the environment for the experiment, like limited food, different food, interactions with the environment? Yes, right. Absolutely.
Absolutely. Yeah. And so that's anytime we as humans are like, so we're kind of trying to, pick what we think is going to be a good phenotype, but it's always important to keep that in mind. Our sort of assumptions or our preconceived ideas about what might be going on here are going to absolutely be influenced by these more complex, like what's going on in the environment, what's the ecology here, all of these things. And so that also fits into this. It's a great point.
We have one from Callum. Dear Nels and Vincent, big fan of the show. I'm a PhD student who is studying and has typically considered evolution in animals, so I find Twevo's consideration in microbes valuable for diversifying how I think about the evolutionary process. I just wanted to suggest some content for the show, a preprint lead by Ben Moran coming out of Molly Schumer's group at Stanford to continue on your interest in speciation and reproductive incompatibility. And this is a paper, a lethal incompatibility between naturally hybridizing species in mitochondrial complex. It's a preprint, actually. I shouldn't say it's a paper.
Yeah, cool.
This preprint tells an incredible story. It identifies specific genes likely interacting to reduce hybrid fitness in naturally hybridizing organisms, making it ecologically/evolutionarily relevant rather than just randomly hybridizing organisms in the lab. They then go on to test what life stage those are acting, what mechanism they be acting through, and the evolutionary history of those genes. There's been interest in the past decade that mitochondria may commonly play a key role in reproductive isolation for several reasons detailed in the preprint. And in my mind, this work is a convincing proof of concept that speciation researchers and all eukaryotic taxa should be paying attention to. Molly Schumer, the senior author on the paper, is a great presenter. I'm sure would be a fantastic guest for the show if you could manage to get her on. Keep up the great podcasting.
This is great. Callum, thank you.
Had you seen this, Nels?
Yeah, this is on my short list. So Molly Schumer's group, so a big fan, and they work on swordtails, the fish that you'll find at the aquarium, like these beautiful, I think they're
Yeah.
live bears, and they have all these great colors and fin types and the swordtail and and then all this underlying very interesting genetics and molecular biology that hit on these issues of what makes one species, like how not just a gene duplication but a species diversification, what makes one species two, the reproductive barriers, and what are the sort of molecular basis of this. Yeah, this is spectacular. And actually that's the question. I think I'll reach out to Molly at some point and hope that she might take the bait and join us. Through her words and descriptions, it would be much more compelling than mine. But I 100% agree. This is another great system being developed as a model system that's unlocking some of these biological secrets, being able to do some of the experiments in this kind of, really cool. Thanks, Callum. Okay, so I think that brings us to our science picks of the week. Kind of excited for the 2 entries that we have here.
We seem to have we're riffing on a theme here, huh?
That's right. So my science pick of the week is DALL-E, DALL-E. This is this artificial intelligence, this machine. I probably shouldn't be careful with how I describe this since I don't know what I'm talking about, but artificial intelligence meets art. So it's been in kind of beta testing mode, or continues to be. These are these great wild algorithms where you type in, a phrase, and then through some seemingly magical process, the computer spits out an image that tries to capture what you're describing. So, and the system has been like, I'm linking to a story from NPR that's describing it much better than I am. And then the actual, you can go on the web now and search for DALL-E, and there are people who have sort of deployed other versions of it. So you can sign up to get access, and I think they've let 1 million people now sort of use their AI algorithm, their engine, or you gain access to it. Other people have figured out ways that you can pour it into it and just do sort of quick experimental stuff like that. So last night I went in and typed in Dictyostelium discoideum with a cat face, and and DALL-E spit out, I think, a really pretty great rendition here. I don't know if you can put this on the screen, but
I can.
Yeah. So we've got here's my science pick, so you can see
hide mine.
The stock cells below.
Yes, down there.
Cat face. I think the whiskers make a pretty respectable. Maybe there's even some fruiting bodies out there at the tips of a couple of the whiskers there. I thought that was pretty great. So anyway, a really fun application of artificial intelligence, which also raises, of course, all of the accompanying philosophical issues as well. But if nothing else, from a sort of practical and fun standpoint,
it's pretty cool.
If I ever need images for like science talks or things like that, instead of hiring a artist to draw something, I think I'll first go to DALL-E and see what the computer has to say about along these lines. Yeah.
Apparently there is a DALL-E Mini crayon, and you can ask for non-politically correct pictures.
Yeah. Hopefully the Dictyostelium with a cat face still falls into sort of polite company. But
this was sent in by a TWIV listener a while ago too.
Very cool. Yeah,
it's very interesting. Yeah, and
really
it will evolve and get better or whatever, maybe worse. Who knows, right? That's what evolution does sometimes.
That's what technology does, for sure. Exactly.
We do stops and starts. So Rima says, this cat can't compete with my Luna. Very nice.
Fair enough. Yeah.
So Rima has a cat named Luna, and I have a dog named Luna.
Yeah.
It's pretty cool.
Very cool. How about you, Vincent? What's your science pick of the week?
So I have a slime mold related pick also. I thought it would be appropriate for this episode. And mine is a YouTube video which are John Bonner's slime mold movies. So John Bonner was a Princeton professor who he actually recently died. I'm gonna share this screen here so you can see John
Okay.
Bonner. Let me see if I can do full screen thingy. Yeah.
Yeah.
He studied slime mold for many years, and he made films of them as an undergraduate. I love the type, the old black and white type there that they're using here. Look at that.
Yeah.
So he made these movies as an undergraduate. And here we go with them fusing, right, to form
Exactly. We're going into the slugs that are culminating into the fruiting body.
And there's music to this as well, which we're
really
not
great.
hearing, but it's really good. It's not very long, and look
Yeah,
at this!
couple
Isn't this
minutes.
gorgeous?
I know these slime molds are absolutely incredible. And I don't remember, I can't remember, if it's time lapse or if it's in real time. There might be some speeding up of the video, but not much. I mean, these things are happening
Yeah,
pretty quickly. Yeah, and then this idea, so that the stock cells will die. There's oh, here, okay, I'll stop talking because we need to watch this.
this is gorgeous. There you go.
Yeah, the building of the stalk. You can see in this case the spore cells are already positioned near the top, presumably with the cup cells on either side. If this is Discoideum, which I think it is. Yeah.
Very cool.
Yeah. Incredible. It would have been fun if, doing the videography of slime molds probably easier said than done, but and there's Bonner.
Yeah, that's John Bonner. Yeah.
Yep.
He died in 2019 at the beginning of the year. He was 98.
Wow.
Yeah. But I followed his career for some time. I don't know how I discovered him years ago, but he wrote a book called Life Cycles. And it's kind of a book about his career and his philosophy about biology and teaching and so forth. It's very nice, very nice little book. And he said, after professors retire, they should be able to remain at a university because they're a resource, right? People can come to them for advice. But very few universities do that, especially medical schools. They want to get you out and get young talent in.
Yeah.
I mean, the week after I closed my lab, I was asked to give up my office, for someone else, which I have a wonderful office down here at the incubator. It's no problem, but it's harder for people at Columbia at least to tap me as a resource, although they can find me online and do it. So I guess it doesn't matter, right?
But it does matter. And so Bonner, for example, taught, I think, for a couple decades after he was officially retired.
He kept teaching, yeah. I mean, I intend to teach in many ways, not just physically at Columbia, but here online. And so I think there are different ways that you can solve the same problem, right?
Exactly. Yep. Yeah, and good reasons for young people to come in and build new stuff, but it's sort of a balancing act. And I think, I agree with you. I mean, medical schools are sometimes a little quick to just sort of move forward without there is no right or, there are ways to do this that are win-win situations. But I think also, what that video and Bonner's story reminds me too is, again, sort of the value of these model systems. So he was taking those videos right back in the 1950s. Here we are 70 years forward, and it's that generational contribution to a system and bringing those tools together. And I think sometimes it's hard, we undervalue that because we don't see it or we don't think about that or talk about it, is just how much work has gone in over decades in some of these cases to really build a community that's given the tools in order to move science forward. And that's part of the story here too.
Yeah, I think in many fields, the past is forgotten, unfortunately. And it's, in dictee, I think people appreciate what was done. Like the paper we looked at today is drawing on very old observations. But often in molecular virology, people forget what was done and they make mistakes as a consequence. And you shouldn't, because if you read the literature, you wouldn't be doing that. And I find too often that many people do not read sufficiently. Someone just started in a lab PhD program and she said, "What's your advice?" I said, read as much as you can, not just today's papers, but go into the old literature and read them and figure out a way to keep notes so you can go back to them, because it'll really guide your thinking. And the old experiments, just because they're old doesn't mean they're bad.
That's right. Yeah.
I mean, they're just as bad as today's experiments can be bad, right?
Absolutely. Yeah. We should be careful. We're starting to really sound like old people. as we discussed.
Well, me, you are not, because I'm the one who is talking here. But I just learned from. I view it as learning from my experience, right? And I want to. I mean, the best thing I can do. In fact, someone here said this. Let me bring it up. Vivek said, "I started listening to TWIM my first year in undergrad. Now I'm getting a master's in applied micro. Keep on inspiring young microbiologists like me." That's exactly the best thing that I can do, right? The best thing I can do is to keep doing that, inspire people. And so when I think back on what I think is important, I say it. So,
yep. Very cool.
All right, Nels, I think that's about it for today, isn't it?
Yeah, this has been fun. I really have to say that paper kind of inspired me here as we're moving into the fall term. And so, yeah, thank you, Vincent, for discussing and kicking it around.
Yeah, it's always fun. And thanks to our members here, 62 viewers who have been contributing to the conversation. And thanks to Les and Steph for moderating for us. And that's TWIVO number 81. You can find the show notes at microbe.tv/TWIVO. You can send questions and comments to twivo@microbe.tv. And if you like our work, you should consider supporting us. You can go to microbe.tv/contribute. You can find Nels Elde at cellvolution.org and on Twitter, @Earlybird. Thanks, Nels.
Thank you, Vincent. Looking forward to the next one.
I'm Vincent Racaniello. You can find me at virology.ws. Music you hear on TWIVO, in fact, I can start it right now, is by Trampled by Turtles. You can find their work at trampledbyturtles.com. Been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for listening. Thanks for joining us. We'll be back next month. Till then, be curious. Bye-bye, everybody.